Why has the conventional drug development system failed Susanna and others? And how is it that Stan and n-Lorem have figured out a way to really adapt this system and be effective? And are there things that we can learn from what n-Lorem is doing, potentially for other diseases? And I guess the final thing is there a point in time, and this goes back to Dan Kern's question, where we might be wandering from nano-rare into ultra-rare? And are we going to see some of the barriers of the conventional drug system come back to haunt us? And if so, are there things that we can be doing today to try to alleviate those? Now, I happen to have a deep allergy to PowerPoint, but I do need one slide for this. And is that up? Yeah. So what happens in a classical drug development process? And you have a graph here. This is time along the horizontal axis, and basically money is on the vertical axis. And in a normal drug development, you first have to start with what is causing a disease. And that's usually work done in an academic institution, but at some point people realize, well, if we could upregulate this or downregulate that or block this, we can actually fix this disease. And then a lot of years of research go into, can we find a molecule of some sort that will actually make that happen? And that's where you have this first phase of preclinical work. And that's what a lot of companies in the Boston, Cambridge area are doing. They're startup biotech companies. It's probably somewhere between $50 and $80 million, and about 5 to 7 years before we actually can test one of those molecules in people. And when we do, we get this thing called an IND, an investigational new drug, and that is basically the assurance to the drug regulators, the FDA in the United States, the EMA in Europe, that this is safe enough to test in humans. And we go into a first phase where we're testing this in healthy volunteers. Then we do this in patients, and we're trying to figure out what's the right dose and are there side effects. And then we go into a third phase, which are usually 2 studies, placebo-controlled, where the FDA is looking really for 2 major things. The first is they really want to make sure as we go into large populations that these drugs are safe. And that's where they use the word rare, because you have to design a study that's large enough to really identify those rare side effects that maybe only 1 or 2% of the population might suffer from. And then, of course, they also want to really make sure that your drug is efficacious and it's working, and you have to test that against placebo. And the point I'm making is all of that costs a massive amount of money, first of all. Just to get to approval is hundreds of millions of dollars. And that's for the one that makes it, because there are probably 5,000 molecules at any one time that are going into human trials for the very first time. But thousands of those will fail. In fact, if there are 5,000 in phase 1, there's only about 30 new drugs every year approved by the FDA and regulatory agencies. So, and that's what really costs an awful lot of money to the system. It's not just the development of the ones that work, it's also the ones that didn't work. And then even then, and the biotech community learned a long time ago, there is something called the valley of death in here. And that is where you get into phase 3 and all of the launch costs, because it's not enough to get a new drug approved. Why is it that Anna had 34 diagnoses, only one of which was correct? Well, part of that is because it took that long for physicians to actually do all the research to figure out what was wrong with Anna. But I can tell you that even for non-rare diseases, it can be a long time before people get diagnosed. And there's a massive effort that is always needed to educate physicians and even to make patients aware of potential treatments. And that's where you get into the commercial launch phase, and that's why you need people around the world who go and help patients. So in a rare disease case, for instance, I talked about Genzyme earlier. I was in Malaysia one day and visiting Genzyme Malaysia. We had 3 people and 10 patients. And you have to make sure that these very expensive drugs are there in the infusion center when the patients come. But we're in Malaysia, and there isn't an infusion center in every street corner. So you actually have to go and help. In this case, I remember one case was a mother with 4 children. Genzyme was there to actually make sure that that mother was able to get bus tickets for her and all 4 children to come to the infusion center, arrange day care for the 4 children while she underwent the infusion, and got the enzyme replacement that was basically helping her lead a normal life. And that's what normal drug development looks like. Now, because of the cost of this, hundreds and hundreds of millions of dollars, when you're talking about the N of 1, nobody can actually afford to do that on the regular system. But the brilliance of Stan and n-Lorem was, well, actually, these ASOs have already been through all that process. You heard about the product Spinraza. Spinraza has been all the way through there. And that gave Stan really the idea, okay, you heard him in his talk this morning say, well, actually, I can use one of these ASOs with confidence because they've already been tested for safety. I need to adapt this so that it can actually fit this particular genetic mutation. But that gives the FDA the confidence to allow that to happen. Now, the one issue we have is going to be what happens. We start with the IND. So n-Lorem is really just working in that first slide of the curve. And we don't actually go all the way to drug approval. And that is great under the foundation model. But what was also interesting was Stan said, well, what happens when we get to ultra-rare? So what happens when we're in that? This model works down to about 5,000 patients worldwide, I would say. We're in a model where we're 0 to 30. What happens between 30 and 5,000? And that's where it gets a little tricky. We ran into that with Qalsody. This is a SOD1 mutation for people with ALS. It's an ASO, also from Ionis. We are charging about $190,000 a year for that treatment. If we had to do cost plus, we'd have to charge a whole lot more. There's just no way that you could ever recoup all of the costs that have gone into the development of this. Because at some point, I think the risk to the model is that we get so many patients and people don't want to stay at IND. They want to see an approval. Dan Curran asked, well, why can't we get insurance companies to pay? Well, the reality is that even the NHS that was talked about on a previous panel, European governments, insurance companies, they want you to get to here. And then we'll reimburse you maybe. But if you're here, you can't. So we will have an education work necessary to say, we are seeing results. We are seeing Susanna stand. How much more evidence do you need? And do you need to go through all of that cost? Because the more cost you add, the less likely it is we're going to have a model that actually is able to finance that. If Stan and John Maraganore tested the foundation, the philanthropic approach to larger patient populations and it couldn't work, I guarantee you it doesn't work. Because Stan is way too much to say he didn't have the time. Stan doesn't do anything except at 300% energy levels. And that's where I think we'll have to think about and educate the system. We have a platform that actually works. We know it's safe. We know that we can adapt it. But in today's world, while we can do N of 1, maybe N of 30, if we're going to get to N of 100, we will have to work within the system. We'll have to get the FDA. We'll have to get insurance companies. We'll have to get CMS in the United States. We'll have to get European governments to say, we actually have enough evidence. The reason that Qalsody cost so much was there are 350 patients in the United States with this genetic mutation. And I listened to the advisory committee. We had families who show their whole family tree about how many people have died of ALS because of this mutation. And yet, the advisory committee is saying, well, we still don't have enough data. And you only get one shot really at doing the clinical trial in something like this, because the minute you show something works, it would be unethical to do another trial. And so I was amazed that the FDA has been hugely supportive. But on the advisory committee, we have people who don't normally see rare diseases. And the mindset of most of the medical establishment, as well as the regulatory establishment, as well as payers, is this model. And that model is the massive evidence base that everybody is looking for. And when you have these small n's, you can't possibly have that. So I just want to put that out there, because as we advocate, I want to first acknowledge really the brilliance of Stan in understanding that he could use this model, and it's his personal credibility as well as the evidence that Ionis and actually Biogen, because Biogen is the one that really did all of the massive investment on Spinraza, for example, but in partnership with Ionis, to actually create the safety foundation, and that Stan has been able to go to the FDA and agree to be used. But at some point, as we expand this, do we run back into that? And that's, I think, what we're going to have to prepare for. So that's all I really wanted to say. I really, I'm just admiring you so much, Stan, for what you're doing there. And you can count on Biogen to help support all your efforts for n-Lorem. And I really want to thank also the Biogen team, Natasha and our heads of R&D, for all the support they're providing n-Lorem as well. So thanks.